Hi students, welcome back to Chapter 3. This will be part 2 of Chapter 3, the molecules of cells. And when we left off in part 1, we were learning about the structure and function of the different types of carbohydrates. Remember that the molecules of cells, the major macromolecules, you can also call them biomolecules. Remember there are 4 types. So let's just recall those really quick. There are carbohydrates, I'll just write carbs, proteins, lipids, and what we call nucleic acids. So in the last lecture, we ended up talking about carbs. And so now what we're going to do is we're going to move on to lipids and then proteins and then nucleic acids. We're going to look at their structure and understand a little bit about their function. So first up, the lipids. There are many different types of lipids. You're most probably familiar with lipids being fats, right? But there are other types too, what we call phospholipids. Waxes are a type of lipid, and then also sterols. So we're going to we won't look at an example of waxes, but we'll look at an example of all the other 3. One of the characteristics of lipids is that they don't dissolve in water. So think about oil and water. You can't mix those 2 things together. Oil doesn't dissolve in water. Oil is a type of lipid. Oils will dissolve, or lipids will dissolve in other lipids, so you can mix 2 different oils together, or 2 different lipids together. So lipids are only going to dissolve in other non-polar substances, other lipids. Functions of lipids can vary quite a bit, but they're a major source of energy. So your fat cells, for instance, have lipids inside of them. And when you're not receiving enough energy from carbohydrates, let's say you go on a low carbohydrate diet, you deplete your sources of carbohydrates, what your body will do is it will then go to your fat cells and pull out the lipids from the fat cells to use those as energy. Remember, carbohydrates, or more specifically, glucose, is the primary source of energy for your cells. That's the source of energy that your cells would prefer to use. But if glucose isn't present, your cells will begin to harvest the lipids and use those as a source of energy. Lipids also can be structural or provide structure. So in a second, we'll look at phospholipids. Phospholipids make up or help give structure to the cell membrane. And then also phospholipids are going to make up the cell membrane. So let's have a look at what a lipid looks like, or a fat, right? So this is the first example, fats. With fats, they come in a couple of different shapes or structures. You'll have a molecule of glycerol right here. You can see that's a 3 carbon chain with hydroxyl groups on either end. And this glycerol molecule has 1, 2, 3, what we call fatty acid tails. So each one of these is a fatty acid tail. All right. So the glycerol on top here in this picture has not yet bonded to its 3 fatty acid tails. Here's the arrow showing that after this reaction occurs, then you have these bonds formed between the glycerol and the fatty acid tails. And if you look at what's removed during this reaction to make way for the bonds to form, it's water. So we've lost a little water. Remember, this is called a dehydration reaction. So this right here then is a fat. and each one of these is a fatty acid tail. Kind of a straight chain like we saw here. That phospholipids are a type of lipid, but they now have a slightly different construction. You can see that the glycerol molecule is shaped a little bit differently. It's not glycerol molecule is again shaped a little different. There's a phosphate group here, and then with phospholipids there are only two fatty acid tails. So you've got these two fatty acid tails here. And what they make are these molecules, these phospholipids become essentially polar, meaning that one end, the glycerol end, becomes hydrophilic. And hydrophilic means water-loving. So one end of the molecule really likes water, and then these two tails right here become hydrophobic. So you know what a phobia is, it means scared. So these tails do not like water. And what happens is you get these phospholipids lining up end to end to end. So see all these heads right here? So you see them lining up like this. All the heads are facing in one direction, and all of the tails are facing in another. And that lines up with another layer of phospholipids below it that's organized in the opposite direction. So here you have the fatty acid tails right here, the hydrophobic tails. So those hydrophobic tails really like to be right next to one another. And then these hydrophilic heads like to be right next to one another, but on the other side. So if this were a cell membrane, this would be outside the cell, and this would be inside the cell. Sorry guys. Okay, so this would be inside the cell, and then what you have on the outside then are hydrophilic heads, and on the inside those hydrophilic heads. But the middle of the membrane is made up of what we call those hydrophobic tails. And this will become important when we start learning about the cell membrane. But for right now, this is all you need to know: the construction of phospholipids, where you find them. They're a main component of cell membranes. So I can go back and let's just draw in, don't laugh at my drawing, I'll just draw in one sterol molecule that might be embedded in this membrane here. I'll draw in another one. So that cell membrane isn't just made up of phospholipids. There are going to be sterols or cholesterols embedded in that. Hey. And then the other type of lipid, these are steroids, sometimes just called sterols. They make up molecules like cholesterol. And oftentimes we think of cholesterol as being this bad thing that collects or accumulates in our arteries, but that's true. But these sterols are major components of membranes. So earlier I was saying that phospholipids make up cell membranes. Well, embedded in those phospholipids are going to be sterols. Membrane also. Not only are sterols membrane components, but they're precursors of other molecules. They're what give rise to things like steroid hormones. These hormones that are things like estrogen and testosterone and progesterone and a number of different hormones that are active in the body. I'm not too concerned that you know the shape of sterols, but on here you can see that a sterol is made of several ring-like structures, and then also a chain hanging off the side, and also there's a hydroxyl group there. I'd be more concerned that you know where we find the cholesterols, the sterols. Oops. Okay, so now we're going to move on to proteins, and I'm going to spend more time on proteins because we spend a lot of time on it in the third unit, and also this is something really good to know, these characteristics of proteins as you move forward in your biology degrees. Proteins have a number of functions, hundreds of functions, but I'm only going to list some basic functions here for you. So let's say, for instance, proteins make up part of the structural network of cells or bodies. So let me give you guys an example as far as the human body goes. Collagen. You've probably heard of collagen. Maybe you take a collagen supplement, or you have a collagen powder that you put in a drink. Collagen is a protein. And in the skin and in bones, collagen acts to give skin structure and strength, but it also gives your bones strength. Proteins, if necessary, can be used for nutrition. So when carbohydrates and lipids are not available, what can happen is the body can utilize proteins as a source of energy, but that's not the preferred source. You wouldn't want to be breaking down proteins in your body for energy because they're needed for so many other functions. All enzymes are proteins. All enzymes. So all enzymes are proteins. Enzymes, remember, are these molecules that help chemical reactions occur. So all the processes that occur inside of your cells or living cells require enzymes for those reactions to occur appropriately. Some proteins act as transport molecules, transporting sometimes sterols. They also can bind with some carbohydrates and transport carbohydrates. Proteins can be used in communication, so they can communicate with other cells, and they're also used in defense or in the body's immune response. Okay, so those are some of the many functions that proteins have. Let's look at the basic structure of proteins. Now, if we look at proteins, or we look at this picture right here, this is a really generic, what we call amino acids. Amino acids are what come together to make proteins. Proteins are usually made up of hundreds of thousands of amino acids. So an amino acid is the individual unit of a protein, or you can consider amino acids as being the monomers of proteins. So many amino acids, many monomers, come together to make these polymers, these long chain proteins. So what you're looking at here is an example of just a generic amino acid and its structure. You'll have an amine group here. So you see that that's NH2. And then you'll have a carboxyl group here. Often that functional group is often written as COOH. And both that amino group, the amine group here, and the carboxyl group, those are functional groups. Those are functional groups that are characteristic of amino acids. Meaning, if you're an amino acid, you've got to have an amine group and a carboxyl group. And then there's going to be a carbon chain. So the authors are just drawing in one carbon atom here for you just to simplify things. But because there are 20 different types of amino acids, it's the rest of the carbon chain that creates the 20 different kinds. So in one type of amino acid, maybe you have 20 carbons in a chain, and that makes it this specific amino acid. Maybe in another amino acid, you have a chain of, 200 carbons. So the R here is sort of a variable, kind of like a variable in math, meaning that the chain off the central carbon right here, it will vary in its the number of carbons. So we call this the alpha carbon right here. Is that alpha? That's a lambdoid. So this is the alpha carbon right here, meaning that that's the carbon that's directly anchored to the amine group and the carboxyl group. And then, of course, this carbon chain here hanging off that alpha carbon will vary in length or number of carbons. So we know that proteins are composed of amino acids. There are 20 different types of amino acids. So in order to create a protein, or in order for protein synthesis to occur, those amino acids need to come together to create a much longer structure. So in this picture, you're seeing one amino acid group. Right here, we'll just call that amino acid number 1. And here's amino acid number 2. They're not bonded together yet, right? But what the authors are showing you is like, hey, here's a hydroxyl, here's the hydroxyl component of the carboxyl group. Here is the hydrogen of the amine group. And through a dehydration reaction, these two amino acids come together. So this hydroxyl group is lost and this hydrogen is lost to create a water molecule being lost. And therefore what happens is you have a bond now between those two amino acids right here. And we call this a peptide bond. Pept means protein. So this bond right here is holding those two amino acids together. So since there are two amino acids, we call this a dipeptide. It's not really quite a protein yet. A dipeptide is really kind of like the precursor to a protein because this is only 2 amino acids long, but really proteins are hundreds of thousands of amino acids long. So you kind of get an idea of how proteins are assembled now through protein synthesis. And that protein synthesis is a dehydration reaction. The bonds that are formed that are holding amino acids together are called peptide. If we look a little bit more closely, we see how complex the structure of proteins gets as we start adding amino acids and creating more peptide bonds. So each one of these is an individual amino acid. There are 9 here, and each one of those amino acids is held together by a peptide bond. So at this point, this molecule is, it's larger than the dipeptide, right? But it's becoming a little bit more complex. It's still in a chain though. It's not long enough for the molecules to kind of start folding back on itself and creating a complex structure. So we say that this is the primary structure of a protein. So we've got those amino acids joined by peptides bonds that form a linear chain, a straight chain so far, that we call a polypeptide chain. So it's a straight line so far. But if we continue adding amino acids onto that chain, the molecule is going to become larger and much more complex. And it's going to take on something called the secondary structure. So this is where we have polypeptide chains folding back on themselves to create sheets and also coils. So here's one polypeptide chain, here's another, and so on. And if we look a little bit more closely, I want to point out that, I'll use black, that these bonds that are holding the sheets together, you guys should recognize those. Those are hydrogen bonds. Remember, hydrogen bonds function not necessarily to hold atoms together. They're not that strong, but they help to create the shape or the structure of very large molecules by creating weak associations. And in some cases, those sheets will begin to create a coil, so you have a slightly even more complex structure. But again, that coiling is due to those hydrogen bonds. So that's a secondary structure of a protein. Now let's move to the tertiary structure. So the tertiary structure now is the structure of a protein where you have sheets and coils packed into functional regions of the protein. So in this photo right here, you've got one coil, another coil, another coil, and another. So you've got all these coils now coming together. And those coils are helping to give this protein even more complex function. And in some cases, you've got areas now of the protein that are going to function a little bit different than the other areas. And we call these functional domains. And not to say that tertiary proteins don't have functions either. Lastly, the quaternary structure. This is the most complex of all, where you have, look at this green chain, that was the green tertiary structure that we saw before. Well, that green tertiary structure will come together with, in this case, a blue tertiary structure to create a protein that's made up of 2 or more chains. So now you have something even more complex. So the reason that these chains are held together so well and so complexly is because you've got those weak interactions, the hydrogen bonds, holding the chains together as one single molecule. So remember what we've been stressing regarding structure and function. The more complex this structure becomes, the more complex its function will become. Most proteins that are functioning, for instance, as enzymes, are going to have this quaternary structure. This is kind of like, you can almost think of this as being the protein's mature structure, where it's actually carrying out its main functions. But many proteins that are fully functioning in the body are going to have this quaternary structure. Now, what if, for instance, some sort of condition or some sort of environment causes these bonds to break? Those hydrogen bonds start breaking. What's going to happen to those chains? Those chains are going to start to lose their structure. You start snipping these bonds, and all of a sudden, the protein loses its shape. And when a protein loses its shape, it loses its function. And that, when that happens, that's called denaturation. When a protein loses its shape and no longer can function in the manner in which it's supposed to. So that denaturation is where that protein unfolds, loses that three-dimensional shape, and therefore loses its function. This can be caused by a couple of different things. Temperature is a big one. So think about having a high fever. Having a high fever, especially for a long period of time and then when it's really high, can cause significant damage to your body. And the reason for that is because most proteins function best, no coincidence, at 98.6 degrees. But if you have a fever, for instance, and your temperature is elevated, let's say 105, that's a pretty good fever, it's a pretty high fever, what happens is those proteins, they're not used to existing at 105. Those proteins start unfolding, and therefore they start losing their function. And then what will happen is you might have death at the cellular level, tissues might begin to die, so you start having damage to the body. And I'm not just talking about humans either. You could think about like fish. Why can't you put a fish from the equator into water in the Arctic, which is much colder? Or vice versa. Why can't you take a fish that lives normally in the Arctic, plop it down in the coral reefs around the equator, and expect it to survive? Mostly because those, let's say for instance, those cold water fishes, let's say they have a body temperature of like 30 degrees. Well, if you plop that fish into water that is, let's say 80 degrees, what's going to happen to those enzymes? Those enzymes are used to functioning at 30 degrees, but then you put that fish in 80 degrees, and the enzymes in the fish's cells are going to go, whoa, hold on a second. We're unfolding. We can't function any longer. So temperature is the main culprit of protein denaturation. This is one of the reasons why we cook our food. We cook meat. And when we do, we're able to kill bacteria and viruses, because that high temperature unfolds those proteins that would make us sick from the bacteria or the viruses. And so this is why cooking your food helps to kill the bacteria, because it kills those bacteria, or it unfolds or denatures those bacteria's harmful proteins. pH is another one. Proteins tend to really like a stable pH range. In humans, our pH of our body fluids, like blood and mucus, mostly blood, should be somewhere around 7.2. A drop in pH of our blood, say down to even 7, or an increase in pH of even just a little bit, let's just say up to 7.4, can also cause proteins to denature. And so can detergents and salts. So think about your laundry detergent. The stains that you've got on your clothes, unless it's paint or something else, many of the stains that you've got on your clothing come from other living organisms. They're caused by molecules like proteins, lipids, carbs, nucleic acids, right? Those are the molecules of life. So if you have a food stain, for instance, on your clothes, and that food was, I don't know, you dropped some chicken on your shirt, and you add detergent to the laundry, you turn on the washer, you've got some hot water there. That detergent denatures those proteins, causes those proteins to unfold and come out of the fabric of your t-shirt. The way that the pH and temperature disrupt the proteins is by breaking those hydrogen bonds and then other molecular bonds that are responsible for the protein shape. So those proteins essentially unfold and lose their structure, and therefore lose their... The last molecule of cells, or the last biomolecule, are what we call nucleic acids. And the monomers of nucleic acids are called nucleotides. So let's look at the structure of a nucleotide first. You can kind of think of a nucleotide as being like an amino acid. It's the building block of nucleic acids. So there are always 3 parts to a nucleotide. There's a sugar. In this case, this is fructose. There's always a phosphate group. And there is always a nitrogenous base. So here's the base in green. Nitrogenous means that it's made of many nitrogen atoms. So sugar, phosphate, nitrogen-containing base. This particular base has the name adenine. And there are 4 different nucleotides that are possible. So this component, the nitrogenous base, can change, but there will always be a nitrogenous base there. It will be one of 4 nitrogenous bases. This phosphate sugar group almost never changes. What's variable to make the different nucleic acids is mostly that nitrogen-containing base. So let's look at the different types of nitrogenous bases. Adenine, you already saw in the previous photo. Thymine, which is often just abbreviated with a T, is another type of nitrogenous base. Cytosine and guanine. And that's in DNA. And so we use these 4 letters, A, T, C, and G, to make up your genetic instruction booklet. These are the letters. So let's say our English alphabet, I think it's 26, I could be wrong, 26 letters in the English alphabet. Our DNA alphabet only has 4 letters. And so the way in which these letters come together is into words and sentences and paragraphs and chapters. They all come together to make your genetic instruction booklet. So in DNA, you'll have strings of nitrogenous bases that may be something like this. And continuing on, another G and a C and an A and a T. And a single DNA molecule is thousands, tens of thousands of letters long. So you could even consider, if you're beginning to write your genetic instruction booklet, okay, well, here's one word, here's another word, here's another, these 3 right here. And so you kind of get how these letters come together to make the story of you, your genetic instruction booklet. So DNA is that genetic information that makes you a human, but also you as an individual. But also in cells, you have another nucleic acid called RNA. And RNA has a similar alphabet. It has adenine, cytosine, and guanine, but instead of having thymine as one of its letters, there's another nucleotide called uracil. So RNA does not have thymine, it has uracil in its place. So the genetic code for RNA, or the genetic alphabet, not the code, the alphabet, is A, C, G, and U rather than T. We'll reinvestigate this as we move through this semester, so these are just the basics. Don't worry too much about learning any more than this for now. In DNA, let's just write, this is part of DNA. DNA, as you know, let me see, 2 sides or 2 strings of DNA. Here would be one. You can see its nitrogenous bases sticking out in the center. When that string comes together with another string of DNA, those nitrogenous bases are going to interact. They're going to bond. So A binds with T, and again, A binds with T, and then C bonds with G, G bonds with C again, and T bonds with A. So in effect, I have it in this picture. Okay, DNA normally exists as this double helix. So here's one side of that helix, and here's another side of that helix. And the nitrogenous bases are here. So adenine, thymine, cytosine, guanine, and so on. And those nitrogenous bases create bonds with one another, like this. And A is only, adenine only binds with thymine, and C only binds with guanine. And when you've got get is this twisted ladder or this helix where the nitrogenous bases are going to bind only with their pairs. There's a really good Amoeba Sisters video on this, and the link is in your Unit 1 folder, and the narrator says, okay, here's a way you can remember the pairs. Apples in the trees. So A always binds with T, and cars in the garage, where C always binds with G. Make sure you watch that video. Make sure you do the worksheet. The narrator there does a really good job of visually showing you in a little animation the structure of DNA. Again, we're going to focus on DNA much more closely as we move along in the semester. And then I'll let you read this slide about DNA and RNA. There is a worksheet, an Amoeba Sisters worksheet and video. It might be the same as the one I was just talking about, that shows you the differences between DNA and RNA. Again, we will revisit these. This is just your introduction to these two types of nucleic acids. DNA is a nucleic acid, and RNA is a nucleic acid. Okay, so that concludes part 2 of chapter 3. If you've got any questions, please don't hesitate to email or come to office hours.